The PFAS problem, a looming threat. PFAS chemicals are everywhere. These synthetic substances have been used in a wide range of industries since the 1940s, and their presence has only grown over time. They're in our water, our soil, and even our blood. The contamination is so widespread that it's almost impossible to avoid exposure. These chemicals are used in many products, from non-stick cookware to firefighting foam. They're also found in water-repellent fabrics, food packaging, and many other everyday items. PFAS are called forever chemicals because they don't break down in the environment. This means they accumulate over time, leading to higher concentrations and greater risks. They can persist for thousands of years. The long-term impact of these chemicals is still not fully understood, but what we do know is alarming. This is a big problem because PFAS are toxic. Even at low levels, they can cause significant harm to human health and the environment. They can cause cancer, immune deficiency, and other health problems. Studies have linked PFAS exposure to liver damage, thyroid disease, and developmental issues in children. We need to find a way to clean up PFAS contamination. Current methods are expensive and not always effective, but research is ongoing to find better solutions. These chemicals pose a serious threat to our health and the environment. Public awareness and regulatory action are crucial in addressing this issue. We need a solution, and we need it fast. The future of our planet and the well-being of future generations depend on the actions we take today. A beacon of hope, introducing F11. Scientists have discovered a new bacterial strain that can break down PFAS. This bacterium, called F11, offers hope for a cleaner future. F11 was found in contaminated soil at an industrial site. This site was in Portugal. The bacteria had adapted to survive in this harsh environment. This discovery is a major breakthrough. It could lead to new ways to remediate PFAS-contaminated sites. Imagine a world where these forever chemicals are no longer a threat. The science behind the solution: breaking the chemical bonds. In the world of environmental science, one of the most challenging tasks is dealing with persistent pollutants like PFAS. These chemicals are notoriously difficult to break down due to their strong molecular bonds. F11 bacteria have a unique ability. They possess a natural mechanism that allows them to interact with these tough pollutants in a way that other organisms cannot. They can break the strong carbon-fluorine bond in PFAS. This bond is one of the strongest in organic chemistry, making PFAS incredibly resistant to natural degradation processes. This bond is what makes PFAS so persistent. These chemicals can remain in the environment for decades, contaminating water sources and posing health risks to humans and wildlife. It's also what makes them so difficult to break down. Traditional methods of chemical degradation often fail to effectively tackle PFAS, leading to ongoing pollution issues. F11 breaks this bond by cleaving it. This process involves a series of biochemical reactions that specifically target the carbon-fluorine bond, rendering the PFAS molecules less stable. This allows the bacteria to eat the carbon in PFAS. Once the bond is broken, the bacteria can metabolize the carbon, effectively neutralizing the pollutant. The fluorine is detached and no longer harmful. This separation process ensures that the toxic elements of PFAS are neutralized, making the environment safer. This process is highly effective. The efficiency of F11 bacteria in breaking down PFAS has been demonstrated in multiple studies showing promising results. In lab tests, F11 removed over 90% of PFOS, a type of PFAS, in just 100 days. This remarkable achievement highlights the potential of using biological methods for environmental cleanup. This is much faster than any other known method of PFAS degradation. The discovery of F11's capabilities marks a significant breakthrough in the fight against persistent environmental pollutants. Section 4. From Pharmaceuticals to PFAS, a History of Waste Management. Interestingly, F11 was not initially discovered for its PFAS degrading abilities. It was originally found to remove fluorine from pharmaceutical contaminants. This previous discovery highlights the bacteria's versatility in breaking down harmful chemicals. The fact that F11 can degrade both pharmaceuticals and PFAS is remarkable. It suggests that these bacteria might be able to break down other types of pollutants as well. This discovery opens up exciting possibilities for bioremediation. It could revolutionize how we clean up contaminated environments. Section 5, Turbocharging Nature's Cleanup Crew Future Research Directions. Scientists are now working to optimize F11 for PFAS remediation. They're investigating ways to make the bacteria work even faster and more efficiently. One promising approach is to provide F11 with alternative carbon sources. This would give the bacteria the energy they need to grow and multiply without slowing down their PFAS consumption. By understanding the mechanisms that allow F11 to thrive in contaminated environments, researchers can enhance its natural abilities. This will pave the way for more effective bioremediation strategies. Section 6, A Cleaner Tomorrow, F11's Potential in Environmental Remediation. The discovery of F11 is a game-changer in the fight against PFAS pollution. PFAS, or per- and polyfluoroalkyl substances, are a group of man-made chemicals that have been used in various industries around the globe since the 1940s. They are resistant to water, oil, and heat, making them incredibly useful, but also incredibly persistent in the environment. This tiny bacterium has the potential to clean up our water and soil, making the world a healthier place. F11 works by breaking down these stubborn chemicals into less harmful substances, effectively detoxifying the environment. F11 could be used in bioaugmentation. Bioaugmentation is a process where specific strains of bacteria are introduced to contaminated environments to accelerate the degradation of pollutants. This process involves introducing bacteria to contaminated sites to break down pollutants. By doing so, we can significantly reduce the levels of harmful chemicals in the soil and water, making these areas safe for humans and wildlife. Imagine injecting F11 into contaminated soil or adding it to wastewater treatment plants. This could be a cost-effective and sustainable solution to a problem that has plagued us for decades. This could revolutionize how we deal with PFAS and other harmful chemicals. The potential applications of F11 are vast, from industrial sites to agricultural lands and even residential areas. The future of clean is looking brighter thanks to the power of nature. With F11, we have a powerful ally in our quest for a cleaner, healthier planet.